be in the test prep.
Hey everybody, it's time for This Week in Science and Education, Personal Plant Project. Hey everybody, thanks for taking the time to join us on This Week in Science and Education. This is your host, Kevin Kugler. Joining me as usual, and in fact, it's my last show, or my first show back since I've been away for a couple of months, so very exciting for me. But joining us as usual, Mr. Colin Jago from the Kawartha Pine Ridge District School Board. Hey Colin.
Hello, hello.
How are things over there in Peterborough?
Well, I'm not in Peterborough. I'm in lovely Brighton, Ontario today, but things are fine. Things are moving right along.
Very good. Well, it's the wonderful thing about virtual communication, because even though Dr. Thomas Merritt is not in Brighton or Peterborough or anywhere, he is in Maine, a whole other country, and he's able to be here. Hey, Thomas, how's it going?
I am well. I'm not sure that Maine counts as another country, but maybe it's still up for grabs on that one.
That's like Canada, right? The 51st, 52nd state.
That's what they told me at the border.
Yeah. Hey guys, excited about the show today. Excited to be back with you guys. We're going to talk to Dr. Laura Hyatt today. Laura is the Associate Dean at Rider University, which is in New Jersey. And she's going to talk to us about the Personal Plant Project and something to do with invasive plant species taking over the world. I don't know, but it's going to be a lot of fun. It's going to be education, so we're glad everybody's with us. Laura, thank you so much for taking the time to be with us today.
My pleasure. My pleasure. So I want to show you guys a picture to start this off. Ready?
Okay.
What is, there we go. Now, when I show my students a picture and I say, what is this a picture of? Guess what they say?
Bambi.
A deer.
It's a deer. But if you look at the picture, there are probably 25 different species of plants in this picture, right?
Sure.
There's trees, there's shrubs, there's ferns, there's mosses, there's all kind of stuff. And students have these huge plant blinders. And so as an introductory biology teacher of a course called Principles of Biology Plants, they sit down in the class and they're not looking forward to it at all. And we start off by thinking about this and saying, you know, hello, there's more here that meets the eye. There's probably 16 times more biomass in a plant in that picture than there is deer. But where our caveman brains are wired to look for food. Now, for food is hard to process and think about. So my goal with this personal plant project, which was featured in Science Magazine as a project for inquiry-based education in the college classroom, is very easily applied in the high school classroom as well. It's all about how plants are perfect starting points for students to learn about how to do experiments and how to trust themselves in the scientific method to answer important questions that are meaningful and teach them something about plants in the process.
So when I was reading the article about the award, my impression is that the focus was really on trying to find creative and innovative ways to get people engaged in the classroom. Now, I understand that you have this whole plant issue that people don't appreciate plants, which is entirely understandable. But as a biologist, I have no idea how plants work.
Right.
But I mean, how much of the course is driven by trying to get students to appreciate plants, and how much of it is driven by trying to get students just to learn the basics of science?
It's very much the latter. It's very much about getting students to realize that we have a method that we use in science to find out the answers to important questions. We're just happening to use plants to figure that out. And along the way, we wind up learning about how we use the scientific method to learn all these things about plants. So they get to the end of the semester and they realize, oh, you know, maybe people aren't the center of the universe. And that, so there's sort of biology fact literacy as well as science fact literacy. And we get the science facts through the process. And so the goal here is twofold, is to teach them some about plants, but it's clear to me most students are not going to grow up to plant scientists. I would love it if they did, but they don't. But what they will grow up to be is consumers, voters, scientists, doctors, and they all need to understand that when information is learned through the scientific method, that there's some serious business there.
Yeah. And that's an awesome.
Yeah, so I hear some of the challenges in that, and I'm just kind of curious, as a non-science guy, I'm wondering, okay, how do you make plants fun? Like, are you taking students out and you're plopping them in the wilderness for a few hours, you're leaving them for a few days at a time, and telling them to bring back all sorts of things? How are you doing this, Laura? How is it fun?
Well, in this particular project, what I do at the very first week is I give them a very basic question and a package of seeds. Okay, so the very basic questions are things like, I read in this garden lore book that I'm not supposed to plant carrots next to dill. Why? That's the extent of the question. They have to develop an experiment with replication, with hypotheses, with quantitative, independent, dependent variables to answer the question about why shouldn't I put these things together. I give them questions about, my seed packet says I should plant basil seeds 4 inches apart. I ask them why. So they design an experiment looking at planting densities, looking at and figuring out different ways to grow those plants. So when they start off the semester, their main memory of growing plants is with putting a seed in a little paper cup in kindergarten, right? Yep. They're convinced they really don't have a very personal relationship with plants. And so the idea when I started this was to have them grow something and ask questions about what we're doing in lecture in the lab. So when we're talking about photosynthesis, they're using a machine to measure carbon exchange in their personal plant and comparing the carbon exchange in their personal plant with other friends' personal plants, right? When we're learning about, kind of what brings me to what I'm doing with invasive species, they figure out what is my plant doing to conduct chemical warfare out there in the universe. And what is it conducting chemical warfare against? Is it bugs? Is it bacteria? Is it fungi? Is it other plants? What's the mechanism there? And so because they have this personal relationship with the plant, they're a little more invested in learning the specifics about their plant, but they're also learning about the process of science in the bigger picture.
The fact that inquiry-driven science is a huge buzzword, right? Both in the States and in Canada, talking about getting kids involved in inquiry-driven projects in classrooms to help understand science. And it's one of the things that is sort of the core focus of a lot of the work that I actually do in my job. And one of the things as I was looking at, and you actually talked about it, the curiosity-driven question that the kids are coming up with. You may sometimes have to start them with the question, but eventually they come up with their own question in their project. But then you talk about tying that in with your traditional kind of lecture knowledge delivery system that universities have, and I'm assuming that it's a fairly standard kind of lecture. So with high school kids, how do you then suggest that we balance, for example, the teaching of the process of science as a method through various things, remembering that we're not just doing one topic, combined with that content, that balance of process and content is something that I think a lot of teachers struggle with. What would you have for advice sort of around that?
Well, so my daughter is in 7th grade, and I'm at war with her science teacher right now. Because
Isn't that great?
Oh, I love it. He's really, really focused on the facts about science. So that poor 7th grade daughter learning about science is flashcards, and then she comes home to her parents who are all excited about, asking authentic questions and thinking about things. I mean, he has them, and then do one experiment at the end of 8th grade in which they do things like water plants with soda to see what happens.
Right. Which they tell them how to do it.
Like, why on earth would anybody do that? What is the question there? I think that, and so I get students in my class who are bored with plants, and they're bored with plants because their teachers were bored with plants, because the way plants were taught for zillions of years was you're going to learn about the petiole, and you're going to learn about all these parts, and you're going to give all these names. And if you don't really like language, and you don't really thinking about prefixes and suffixes and how words communicate to you, it's going, nobody cares. So I would, and my 3rd grade daughter, who is being taught science by somebody who really doesn't have a lot of command of science facts, really gets the scientific method. Because, they're doing silly soap and eggs and, materials and candles, and it's not about, fire and flames and temperatures, but it's about why does this happen and how can we figure out why this happens. And so, Richard is an engineer, and he has them building has them build like bridges, has them do all the sort of traditional like build a robot, build this, that, and the other thing.
Right.
But there's no, I mean, engineering is great, but that's science. I mean, it is and it isn't. Engineers would hate me for saying that, but so I would actually encourage even more soft peddling of the facts at the high school level. I mean, I get college who say like, when are we going to dissect? Because that's what science is, dissection.
Right. Yeah. As opposed to what are we learning and what value would a dissection be to a question that I have to answer, right? They jump to the thing that we've always done versus having a question that actually drives what you want to do.
Right. So, for instance, when you're teaching about phototropism, right, there are all these classic Darwinian experiments that they did to learn about phototropism, about signal transduction and auxin and all of this other stuff. And it's a whole series of experiments that I spend like 3 days going through. Now, I'm really, by the end of it, students know about auxin, they know about, transit, they know about fat-soluble and water-soluble compounds. But what's more important is that we talk about how you design the experiment and what the hypotheses are and what kind of outcomes would we expect on the basis of other things. So it's, yes, they learning about why plants bend towards light and how plants bend towards light. But in the process, they're learning about how we got to those facts in the first place. And I think maybe more focus on fewer facts, but the mode of acquisition of those facts would be much more helpful in high school. And you could, cover three facts in all of the high school class, and it would naturally take you to other places. I'll shut up, Thomas.
No. I mean, that's awesome. And we've been saying very similar things, but honestly, with less from the scientific from the scientific perspective, less of an educational perspective than you have because of this course that you've developed. But I mean, isn't one of the difficulties that we run into is that because things are so exam based, that you have to give the students lists of facts, not because you want to or because they're going to learn more, but because they're going to be evaluated nationally, locally by these fact-based exams. And it's really freaking hard to make an exam that tests a student's ability to creatively solve a problem. It's not that it can't be.
It is assessment, trying to figure out, did you do it? Did you get it across to these students? And actually what I do at the end of this class, one of the final questions is, again, it's pretty divorced from telling me about specific facts, but I say, okay, we go on a field trip at the end of the semester and we walk around our campus and we look at patterns. And I tell them this in the last class. I say, your final question on your final exam will be design an experiment to explain a pattern that you see out here in the woods. They know they're learning a little bit about the species. I give them a little stuff, but what it really is testing is, did you figure out what replication is about? Did you figure out what controls are about? Did you figure out about how we make tests? And sometimes they make, ridiculous hypotheses, but their designs are beautiful. And that's all I want, Sometimes the hypothesis is, aliens came here and planted these trees in this configuration because they're communicating with the mother planet. And so then they come up with an experiment to test or refute that hypothesis. They tell me what results would you get if it was supported? What would you get if it wasn't? What would it look like in a graph?
Right. Yeah.
So more questions like that at the school level would be awesome.
Yeah. Mm-hmm. And it's embarrassing at what frequency I see fourth-year university students defending an honors thesis, and they have no idea why they would have replicated that experiment. And it's like, somebody along the line has screwed you over in your education. I mean, you should understand replication, experimental design, but, oftentimes you can get through your entire education and never be challenged with that.
Absolutely.
Especially if you study molecular biology. I'm married to one.
Well, let's hope you're not watching this podcast after the fact. Laura, I've got a quick comment for you. I have a suggestion for you, actually. A couple of years ago, the three of us went to the Canada Science and Technology Museum, and one of the things that I brought home was a rocket ship that you can, fire off into space, right? Little mini one. So I came home and I talked to my 2.5-year-old at the time about, jet propulsion and all that. And of course, he had a blank stare. But once we set off that rocket and then we subsequently blew some stuff up, he was right into it. So, inquiry-based, hands-on, all that kind of stuff. If you could figure out a way to blow up some plants, you would have like this whole other student engagement that you wouldn't even know about. So I'll leave that suggestion with you.
Well, one of the things that I do in this class is I have a food of the week. So I make students eat kumquats. I make them eat edamames. I make them eat drink spirulina shakes. I use I call this tongue as teaching tool. So, maybe they're not exploding plants, but they're ingesting them and they're thinking about the nutritional value in them and where the seeds came from and where's the flower. And, we eat saffron crackers when we're talking about stigmas, and it's very easy to make those connections for kids, and you always have to be looking for something that's going to grab somebody and make them start asking questions.
Yeah,
absolutely. We have to do the same thing with Colin all the time. Sorry, Colin, you had a question. Go ahead.
that's okay. You did it nicely that time, so well done. As I was reading your stuff and as I'm listening to you, it strikes me that what you're doing is what everybody should be doing. So my question is, how unusual is your approach? I mean, you won this award for the Personal Plant Project. How unusual is your approach within your university and maybe broader within your discipline in universities in the U.S.?
I would say there are a couple levels to that question. The first is the reason that I do personal plants the way I do is because that is a lot of the way we teach science at Rider.
Okay.
I have a lot of who are really talented in getting students to devise authentic experiments to answer really authentic questions. I'm standing on the shoulders of giants, right?
Awesome.
I would say, so it's very prevalent there. We also are the host of the Bristol Myers Squibb Center for Science Teaching and Learning here, which is very much centrally dedicated to increasing the frequency with which New Jersey and Pennsylvania science teachers use inquiry in their classroom. So there's a lot of scholarship and activity about that going on here at Rider, and I think it could be spreading further. I don't think botany is taught this way because botanists have not been taught this. I was not taught this way.
Right.
I know more arcane plant words than you can imagine, and when students come into botany and they think about classification. They want to be able to identify a flower. Like, that's what they think botany is. They don't think about plant-pollinator interactions. They don't think about the roles of plants in the carbon cycle. They don't think about the roles of plants in creating building materials, any of that. So I wish there would be more of this in the botany world.
Are you challenged?
Sorry, I was just going to point out we've got about 2 minutes left, and it's usually my custom to pick on one of these guys for the last question, and Thomas, I'll pick on you today. Last question of the day, please, sir.
I have just a really quick question, and then I have a better question. The quick question is, a lot of times when we talk to people about this style of teaching, the first point that I brought up with, you've got to teach to exams comes up. And the other thing is it's tough to teach to groups of students. So how big a class can you effectively run with this approach?
I had 86 students the last time I taught this. But they are in classes, they are in labs of 16.
And there is no word that we actually will allow on this podcast that will fit the exclamation that I was going to use when you said 86. That's awesome. That is not the answer I thought you were going to give me. So you've taken a big class and you break it down into smaller units, and that works fine.
Yeah.
Okay, my last sort of more scientific question. I mean, you have a personal interest because the points that you brought up at the beginning, which were awesome, about just a better educated population. Have you found a way, given all the time that you've had to put into making it an innovative way to teach, can you tie your own research into what you're doing?
Great question.
Yes, that's really fun. Actually, some of the questions that I give students to do are connected with, and my research has gotten much more involved with allelopathy and involved with how plants conduct chemical warfare underground, because that's what my students really want to do. They want to use an HPLC and measure the concentration of synigrin in the ground. A lot of plants squirt secondary compounds in the soil that can then be absorbed by activated carbon. And so I use garlic mustard, which is the species that I work on, as a model species for them. It's kind of hard to germinate garlic mustard because you have to cold stratify it for 3 months. But what we do is I get students, I give them the gateway drug of connecting other, like that carrot-dill question, that interaction, that negative interaction between carrots and dill is driven by a compound called furanocoumarins, which is very common in the Apiaceae, which is a plant family that they both belong to. Dill makes it, carrots don't. And when you grow them with activated carbon in the soils, the effect of dill on carbon is much mitigated because the activated carbon is absorbing the furanocoumarins and it's preventing that interaction. So then the only interactions you have are shading interactions instead of chemical warfare interactions. So, even though we're doing density experiments, we're doing crop rotation experiments, there are these other sorts of entrees to further investigation of invasives and stuff like that.
Laura, do you know what's been great about this show is that Colin, Thomas, and I have been talking over each other the entire time, which I think is a testimony to how interesting you have been. It's just great. So on behalf of these characters, I just want to say thanks so much for coming on the show today. We really appreciated talking to you. And we hope you had a good time, and maybe you'll come back again and talk to us in the future, because I know we've got lots more questions.
I would be happy to.
Thank you, Laura.
Thanks again to...
All right,
Sorry,
thanks.
go ahead.
No, I said thanks.
Thanks again to Dr. Laura Hyatt. She's been our guest today on This Week in Science and Education. And you can find out some more information about her by going to her website, which I have right in front of me here, Laura. I believe it's, I'm not sure which one you'd like me to give, but www.rider.edu/faculty/Laura-Hyatt. And, of course, you can always go to our T-WiSE website at vrock.ca and get some more information and some links to Laura as well. Laura, thanks again, and thanks to everybody for tuning in for another episode of This Week in Science and Education. Have a great week and a Merry Christmas, everyone. Take care.